I'm a professor in Cornell University in the Department of Chemical and Biomolecular Engineering, and today I'm going to be talking about really how to design and some of the things that I think folks should think about if they're interested in getting into this space of antibody-mediated RNA delivery. And for the most part, actually for all the part today, I'll really be talking about just antibody-siRNA conjugates for delivery. Now, you can make antibody-RNA conjugates, antibody-DNA conjugates for a variety of immunotherapy applications, but for our session today, I'll keep it focused on the delivery of short interfering RNAs. I'll go over techniques related to antibody structure and function, in case there are folks here that aren't familiar with the structure of antibodies. I think everyone must have heard of what an antibody is, but there's salient features of it that I think are important to take note of when you're thinking of putting together some sort of antibody-RNA conjugate. Then I'll go into why I think, and I think a lot of people do think, targeted RNA delivery is sort of the perfect therapeutic modality. And I put a ... there, and of course that's if you can make it work, right? So there are issues with delivery and there are issues with escape that if you're familiar with this field that you have to deal with. But the concept of making one specific conjugate that you can use to target another very specific macromolecule is of great interest to the field. And there are a number of industrial folks interested in this topic as well. I'll go slightly into some antibody conjugation techniques, how you put some of these things together. I think conjugating them and putting them together is perhaps trivial. Anyone can sort of add one thing to another, but I think characterizing and understanding what you have and understanding what your degree of labeling should be and your drug to antibody ratio should be, I think that's where it gets difficult. So we'll talk about some techniques that my group has been involved in, but also others involved in making some of these antibody-RNA conjugates, and sometimes you'll hear them being called ARCs, ARCs. Right? So ARCs, the quest for escape. I'll then go into a number of examples in literature that folks have used to make these conjugates and what they've done and what the promise is and where we should go from there. This field of antibody-RNA conjugates is actually not that big. There are really only about 10 to 15 key papers ever since 2005 in this field, just to let you know of how difficult it is to make these conjugates. You can contrast that with polymeric conjugates or lipid conjugates, where you would probably get 15 papers on that topic every 2 weeks. Right, so it's a difficult space to get into. If you think about it, you're trying to conjugate or take one really complex molecule, antibodies, it's not easy to make those, with another really interesting and complex molecule, short interfering RNAs, right? So we'll go through this as sort of an outline and we'll sort of discuss what some of the salient features are, and I think what folks really need to think about if they're interested in getting into this space and how to sort of analyze some of the results that come out of it. All right, so an antibody is a Y-shaped molecule. I think everyone is familiar with what an antibody is. It has binding domains. It's always drawn in this format. You have your binding domains up here. It's really a dimeric macromolecule. You have Fc regions here, and you have a light chain over there. And again, it's drawn as this simple Y-shaped molecule, but it's a lot more complex than that, right? So if we really look at the antibody structure, things that are really not drawn for the most part, and I apologize for folks sitting on this side of the room, but I can only point at one screen at the same time. But I'll try and talk through it as well. But again, it's a more complex molecule, right? So if we just focus on one half, it's a dimeric molecule, so everything you're seeing on the right-hand side of this macromolecule is also present on the left-hand side. You have two regions of your antibody. You have a heavy chain and you have a light chain, right? The heavy chain is this green and blue region here, and you have a light chain over there. Your antigen binding domain is a combination of the variable chain at the tip of the antibody and the variable chain on the light chain of the antibody. So you have a variable chain at the tip, you have a constant region for your heavy chain, you have a variable chain at the tip of your light chain, and you have a constant region of your light chain. Something that will be important as we go through this talk to focus on is just the molecular weights. The whole antibody is about 150 kilodalton in terms of its molecular weight. Each half, this half that I'm showing here, is about 75 kilodalton. The heavy chain itself, just this chain right here, is about 50 kilodalton and the light chain is about 25 kilodalton. It's important because when I show gels regarding how you conjugate things, I'm going to be showing reduced gels. The reduced gels means you're cleaving this disulfide in the middle, so you're only generating a half chain. So on the gel, you'll either see a 75 kilodalton band corresponding to this whole thing. If it's completely reduced, there are also disulfide bonds between the heavy and the light chain in here. They're called intra-chain disulfide bonds. Those are also cleaved, right? So in the gel, what you'll see is a 50 kilodalton band, and that 50 kilodalton band corresponds to the heavy chain, and the 25 kilodalton band corresponds to the light chain. It's important because if you're doing site-specific conjugation, these salient features in terms of where you conjugate to will be important because you can actually see those on a gel. Right, so if you see your 50 kilodalton band move, you know you're conjugating to the heavy chain. If you see your 25 kilodalton band move, you know you're conjugating to the light chain. If you see everything move, it's a non-specific conjugation. All right? Something else that isn't drawn when you take a look at antibodies, or they're drawn this way, is the glycosylated regions. Right? So most of your IgG1s are glycosylated, so you have a secondary response region shown here that's extremely important. It's going to be ignored as well for the most part of this talk, but whenever you see an antibody in this talk, there's always a glycosylated region on it. This is a really nice handle, and unfortunately, I won't have time to talk about it today, but this is a site-specific handle that you can actually deglycosylate and conjugate specifically to the Q295 amino acid right there. So you can actually use it for site-specific conjugation. If you're interested, there's a technique known as, again, using PNGAs to deglycosylate and using an enzyme called MTG to actually conjugate specifically only to the FC region of your antibody, which is extremely desirable, right? You want to be able to conjugate down here and not conjugate up here in your antigen binding region. This is what's required, or this is what folks want for recognition. All right, so that's what the antibody structure looks like, and we're going to be conjugating to the antibody. We're going to be conjugating RNAs to the antibody to be able to generate these conjugates. Now, antibodies themselves have a diverse array of functions. They're involved in antibody-dependent cellular toxicity, shown here, where they bind to specific receptors on the surface of your cell, recruit natural killer cells, NK cells, which then either phagocytose sort of the molecules of interest, or are able to destroy it by a variety of mechanisms. You also have CMCs, this complement-mediated toxicity, where you recruit complement to the surface of your cell, which causes lysis of your cell. And of course, what most people here are interested in are making antibody-drug conjugates that have been around for over 2 decades now, in which case you're conjugating some sort of a cytotoxic drug to your antibody. Your antibody mediates binding to the receptors on the surface of your cell. This undergoes endocytosis. If you're clever about designing the linker between your cytotoxic drug and your antibody, you can target this for site-specific cleavage within the endosome via some sort of enzymes, for example, CAT-B, that will mediate release of your cytotoxic drug into your cell and mediate action. Right? So this is what we want to achieve, or this is what we would like to achieve with RNAs. Right? Piggyback, or at least take the lessons from antibody-drug conjugates, where you're conjugating some sort of cytotoxic chemotherapeutic drug, but you replace that with short interfering RNAs, and perhaps you can target this to any cell type you want by essentially just raising an antibody against that cell, mediating uptake, cleaving, and somehow magically escaping the endosome, and of course having function, right? Magically escaping the endosome, that's the key word there. All right, so how do we do this? This is sort of the premise. This is what we would like to do. And again, most of you are familiar with this field. This has been spurred, I think, mostly, again, by the system shown here. This is the triple GalNAc system from Alnylam. It sort of represents, I think, what the hallmark of what antibody drug or antibody RNA conjugates want to be. Right? So this paper in JACS was published in 2014. This work was done way before then. But here they're taking heavily modified RNA. So that's another thing you have to do. If you're conjugating an RNA directly to an antibody, this is not in a nanoparticle. Your RNA needs to be heavily modified in order to be able to withstand the barrage of nucleases that are going to be thrown at it. So for example, if you take a look at the RNA shown here, the dots here are phosphorothioates. The italicized capitals are fluoro modifications, and the small letters are 2'-O-methoxy modifications. So this is a heavily modified RNA. to be able to impart nuclear stability to this macromolecule. Conjugated to this, of course, is your triple GalNAc system that targets the acetylglycoprotein receptor on the surface of hepatocytes, binds with nanomolar affinity. This is a direct conjugate to your RNA. And this system is being well designed. You get about 50% knockdown at about 1 mg per kg when you do a subcutaneous delivery. This doesn't work as well for IV delivery. And we can go over why that is. But the way it functions, again, is you're engaging some sort of a receptor, the acetylglycoprotein receptor, on the surface of your hepatocyte cell. This is for liver delivery. You undergo endocytosis, and then you traffic, and somehow, magically, your RNA comes out, and it does function, right? And I go magically, right? So what do you need for this to work? There is some element of endosomal escape that has been postulated that occurs in a variety of endosomes. It's not clear what cells this is most active in, but some of the things that I think you'll see as we go through this talk are things that you have to pay attention to if you do want to be successful in building some of these conjugates. This is going via, again, this acetylglycoprotein receptor pathway. This is a unique pathway because you have a super high expression level of these receptors on your cell. And by high, I mean north of 10 to the 6 receptors per cell. That is pretty high. That's almost similar to what HER2, the number of HER2 receptors are on the surface of, say, SCOV3 cells, for example. For the most part, receptors on the surface of cells, you're looking for things in the 10 to the 4, 10 to the 5 region. 10 to the 6 is super high. So you have a super high expression of these receptors on the surface of your cell. You get rapid uptake, so endocytosis is rapid, so you get rapid uptake of your conjugates into your cell, and you also have fast recycling. And it's thought that it's this high expression level combined with the rapid uptake that facilitates endosomal escape. So if you believe in the stochastic process, and the belief is that you do have random escape happening, these endosomes are leaky, it's not high, it's probably less than 1%, but that gets made up for because of the high receptor expression and the high uptake into your cell, right? As we'll see, it's not a universal concept, but it does hold in some cases. Some other people have found this out to be true. And of course, the goal here is, can we find other systems that do this? This is going to the liver. This is present on hepatocytes. There are many other targets than liver targets that folks are interested in. So the idea, of course, is if you can replace this triple GalNAc here with some other binding entity that binds with similar nanomolar affinity. You could do this with antibodies. We know how to make antibodies. We know how to express antibodies. We can essentially make antibodies against anything we want. And you can piggyback on this process or find it in other cells, then perhaps you can get this working, right? So what we'll talk about is how to do that and what the promise looks like. All right, so ARCs. Here's what we want to do. Take an antibody, conjugate an RNA to it, an siRNA to it with some sort of cleavable bond. Maybe it's cleavable, maybe it's not. We'll talk about that later today. In fact, I think cleavable is not the way to go when making these kinds of conjugates. Have it undergo endocytosis. Again, believing that there is some level of endosomal leakage, right? When endosomes undergo homotypic fusion or heterotypic fusion, there's some element of leakage that goes on, and that degree of leakage is cell-dependent, right? We don't know what it is, but it's cell-dependent. But if we can target cells with a super high expression level of receptors, again, north of 10 to the 6 per cell, target cells that undergo rapid uptake, perhaps have fast recycling, perhaps we can get this to happen as well. It is unclear whether or not, what happens in this pathway. This is going to be the story for today. Do you have escape in the early endosomes, in the late endosome, in the lysosome? Is your RNA getting cleaved and just coming out by itself, or is it coming out with the whole antibody? No one really knows, right? It's not really clear, but there are things here that I think we need to understand. And there are a lot of players that are interested in this, right? I do know that Avidity Biosciences do have a product, an RNA antibody drug conjugate product that they're pushing forward. Roche and Genentech were interested in this, at least they were back in 2014 when they published a nucleic acid research paper on it. We'll go through what that paper detailed. And also based on the talk by one of the Amgen representatives from the PEGS meeting a couple of weeks ago, they're also interested in this space. And I'm sure there are many other companies interested in this space as well, because the potential is high, right? It's a single conjugate. From the FDA, this would be appealing to work with. It's well-defined. It's not a heterogeneous product. And again, if you can get this to work, you can design a platform out of this, because you can then just go picking different antibodies and different RNAs and making different conjugates for different diseases, right? So the promise is high. It is difficult to build, though. But this is what we want to understand. All right, so my lab mainly actually works in antibody drug conjugates. We make chemotherapeutics. We're interested in things like being able to suppress aggregation of those chemotherapeutics. With antibody drug conjugates, with the drugs, the chemotherapeutics, and small molecules, the concerns are a little different because those molecules actually do cross the cell membrane. They're highly hydrophobic, so you have to worry about things like aggregation. But I think the concepts do cross over. When you conjugate your drug, be it an RNA drug or small molecule drug, really doesn't matter what it is, you have to be cognizant about what we talk about in that field are drug-to-antibody ratios, so DARs, or degree of labeling, DOLs. You'll find that often in text. And your drug-to-antibody ratio, again, as the name suggests, talks about this. If you're conjugating non-specifically to your drug, especially with something that is hydrophobic, you typically lead to aggregation, liver metabolism, and you decrease your efficacy of your product. Again, if you're conjugating with a linker that isn't stable, you get premature release and you get a circulating cytotoxic drug, which you don't want. In the case of RNAs, perhaps you don't get aggregation, but you surely will get this. If you over-conjugate with a high DAR, that is a high drug-to-antibody ratio, you get poor receptor engagement, poor binding, and low uptake, right? If you under-conjugate, again, perhaps you might not see anything at all. So you really want this conjugation to be as precise as possible. Preferably, you want to bind or conjugate not to the binding site, so site-specific methods are preferable, though not prevalent in this field at the moment for understandable reasons. But you want to be able to create a DAR of about 1 or 2, right? Where you're conjugating really to the FC region and not to the Fab region or the binding region of your macromolecule, which should facilitate uptake into the cell. So there are many ways of doing this. There's the site-specific methods and there's the random conjugation methods, or at least I'll call them statistical conjugation methods. We'll go through the site-specific method first. This method is not widely used for siRNA conjugates. It is widely used for chemotherapeutic conjugates right now. That's a much older field. But this essentially involves a variety of methods, right? So here, you have to be able to make the proteins in-house. So these are recombinant proteins where you can include, say, an azido amino acid anywhere within your sequence, but there are hinge cysteines that you can take advantage of. There's selenocysteines. There's this formoglycine method where you can include a cysteine within a specific 5-amino acid region in your sequence, and using this formoglycine enzyme, you can turn it into an aldehyde, specifically anywhere within your macromolecule. Catalan Biologics are actually involved in this with David Rabuka. But then there are also tags you can include when you make your antibody. One that we've used in our lab are lab tags, right? So you can include a lab tag. It's this lipo acid ligase that conjugates this 13 amino acid. You can put this 13 amino acid anywhere within your sequence. Here I'm showing it at the end of your heavy chain, but you can also put it in your light chain. And then with a specific enzyme, you can conjugate a substrate to it. Here you're conjugating specifically to the lysine and attach your drug in a site-specific fashion. But you can also do it with sortase tag or butylase tag, and then many other tags that you can use to do this in a site-specific fashion. Just showing you just how well this works. This is a targeted conjugation. So here we're just conjugating pegs, just as an example. But here's that 50 kilodalton band that corresponds to just your heavy chain, shown here. So if your lab tag, for example, is on your light chain, here's your light chain, here's your heavy chain. You see specific conjugation to your light chain. You see no conjugation to your heavy chain. Highly specific. You can do the same thing. You can put your lab tags on your heavy chain, conjugate to your heavy chain. You see no difference in your light chain because you're not conjugating to it. But you see a move up to your heavy chain showing that you're only getting heavy chain conjugation. So these are very specific methods that you could use for these conjugations. Now, in the field of siRNA conjugates and the examples that I will show, these methods are not being used. Again, these are rather new. They're being used for antibody drug conjugates with chemotherapy. But for siRNA conjugates, pretty much all the conjugations, except for the ones where they express the proteins themselves, are done via these random conjugations. Now, the field of antibody-RNA conjugates is sort of a catch-all term. Not everyone uses an antibody. In fact, most people will use this Fab prime fragment. Some people will use a Fab fragment. Some people use a single chain variable fragment. These are all fragments of your antibody. So this is the Fab prime, including this hinge region. The Fab is just this region. The single chain variable fragment is just the tip of your antibody. They're all sort of under the umbrella of antibody drug conjugates. So again, if you're seeing scFv conjugations, and they call them antibody-drug conjugates. It's essentially the same thing. So here, most of what's been done is using the lysines that are present, the external lysines that are present on your antibody for conjugation. And the way you do that is you essentially take some sort of NHS drug or some sort of NHS drug with a cleavable bond in it, where your drug could be your RNA, and you directly conjugate that to your antibody. It's a non-specific method. Your drug or your RNA goes everywhere, right? And you can tailor this in order to minimize reduction unbinding. Another method folks have used is you can activate your antibody with Trout's reagent to give you thiols on the surface of your antibody, and then use this non-cleavable SMCC linker to attach things to your antibody. So unlike the gel that I showed before, looking at the gel at the bottom here, here's your 50 kilodalton band, and you see your smear of bands, right? You have 1 conjugation, 2 conjugations, 3, 4, and so on and so forth. It's non-specific, which might be undesirable, but it does work. It is fine. You can tailor this by tailoring the stoichiometry of your conjugations, and you can actually achieve just 1 to 2 drugs per antibody if you're really careful about your conjugations. But again, these gels show the difference between doing a site-specific versus doing a random conjugation when it comes to attaching drugs to your macromolecule. Here's an example of something that we've done in our lab, and there are many examples of this, but these are just things that I think you should be aware of and take into account if you're trying to make these conjugates. A lot of these characterization techniques, I have to admit, are not seen in papers. In fact, you would rarely see some of these, and I applaud the papers that put them in because I think they're highly valuable. But here's an example of a non-specific conjugation. So here you're taking an antibody, you're conjugating it to this tetrazine NHS, so this is your activated end. This reacts with lysines on the surface of your antibody, so you put tetrazines all over. I'm only drawing one here because we've done a lot of work here, and we know how to put really just one, even if you're doing this statistically, and that data is shown here, right? So this is the equivalencies of tetrazine that you're adding, and you can really dial the conditions and the buffers that you're using such that your degree of labeling shown here that you can detect from SEC or other methods really is just 1. Right? So we really are just putting 1 tetrazine per antibody, right? So there are ways to do this by engineering the stoichiometry so that you do only get 1, even if you're not using site-specific chemistry. And then taking that 1, you can take your RNA, label your RNA with a TCO modification, and undergo this inverse Diazold-Demand addition to put your RNA specifically on your antibody, and then you can hybridize with the antisense strand to create your antibody-RNA conjugate, right? If you want this to be cleavable, you can include a cleavable bond right there if you want it, right there, sorry, right here if you want. And we've done this using a variety of methods. So again, here is showing that you are only really putting 1 tetrazine per molecule. This is a really nice characterization. You can actually characterize these things really well by size exclusion chromatography. If you're interested, I've sort of given the column that we use that works really well. Here in black is your antibody before modification. In red is actually a singly modified antibody with a tetrazine. And you might be like, whoa, how can you detect this really small molecule by size exclusion chromatography if you're only adding 1 of this, which is about, what, 0.5 kilodalton to 150 kilodalton antibody? There's actually an affinity effect going on here. So this is not solely based on size. These things are actually adhering to the surface of that gel. But it does work. We've characterized this by MALDI. This is the single conjugate. If you take the single conjugate and you do this reaction and put your RNA on it, again, shifts to even a bigger peak. So this is your pure, singly modified antibody-RNA conjugate. And again, you can verify that by doing MALDI on that peak as well. Whoa, I am running widely out of time. Okay, here is the gel, antibody-RNA. This is a native gel, so it's running down because it's more negative this way, so it's not weird. This is your conjugate. If you look by Cybergold, your RNA is right there. So it's a really nice way of controlling stoichiometry, making these antibody-RNA conjugates. Again, without going to expensive site-specific methods. Do these things work? Yes, if you hybridize with a fluorescently labeled antibody, this is done with transferrin, CD63, HER2 in my lab. You can see internalization in all the cells. You can see binding, again, within about 2 or 3-fold of what the parent antibody looks like. So again, you can make these things quite efficiently, quite pure, and then be able to do studies on them. I'm going to go a little bit quickly because I want to get to the punchline of the story. So again, ARCs, antibody-RNA conjugates, mean different things to different people. I call them antibody-RNA conjugates, which are antibody-RNA conjugates. ARCs are also referred to antibody-RNA complexes, right? So actually, the majority of stories that you'll see today are actually antibodies conjugated to cationic polymers or sequences, and then your RNA is complexed to those. Those are also called ARCs. Those are antibody-RNA complexes, not antibody-RNA conjugates. It's a subtle difference, but it is a difference. I don't consider these single conjugates. Why? Because I think ultimately these will form a nanoparticle. This is the equivalent of taking a polycation, mixing it with the RNA, forming a particle, and then conjugating your antibody to it afterwards. There's nothing wrong with that. But here you're just doing it inverse. You're taking your cationic agent, conjugating it to your polymer, and then the RNA, and I think you have an equilibrium going on between these two, right? Both called ARCs, but there's a subtle difference. Just take notice, these are actually direct conjugates. These involve your polycations. I think this is the way to go. Ultimately, you will see very good data on these systems, though. I think these systems will still suffer from dissociation in serum and so on and so forth, but they actually do work, and they do work quite well. The conjugation techniques I talked about that apply to RNA conjugates also apply to conjugating a protein on the surface of a polymer on the surface of your antibody. Okay, so this paper by Judah Lieberman and Eroy Song was sort of the first system here, I think, to make these antibody-RNA conjugates in 2005. There might have been earlier systems. If there are, please let me know. But this was the Fab protamine conjugate. This was actually expressed as one conjugate, so this is not a synthetic conjugation method. This is protein engineering expressed in this whole system, adding your RNAs to it. And again, here they're showing very good binding between the RNAs and the polymer here. The polymer here is protamine that they are binding to. Data here again shows that you see very good binding in the presence of cells that have this GP160 protein on the surface of them. And again, they were able to show pretty good in vivo data. Actually, this is intratumoral injection, and this is IV injection. Again, showing tumor remission. Again, showing that you get RNAs at the site of your tumor. So again, these systems seem to work. And in all these slides, I've sort of summarized by putting what the rough efficacy was. So this was delivering about 4 mg per kg, 3 injections per week shown here. And there are many systems that have done this. So I'm going to summarize this all in one slide because these are all antibody-RNA complexes, not antibody-RNA conjugates. But 2 years later, David Pierre had this paper where here they're taking single-chain variable fragment protamine expressions. Again, these are all expressed, adding the RNA to it. They show efficacy in vitro. They don't show any knockdown in vivo, but they show co-localization in vivo with tumor cells. A year later, Cell paper, again, this is an SCFV, the tip of your antibody, single-chain variable fragment, protamine. This is a 9-arginine. This is actually conjugated to your single-chain variable fragment by some of the techniques I talked about. 2 mg per kg injection in vivo, 80% knockdown shown in that system. And again, the list goes on. Here's an interesting system targeting HER2. This is a single-chain variable fragment. Again, conjugated to protamine, it's a complex, and the list goes on. Right? So again, these are antibody-RNA complexes. The first paper that I think really got at making antibody-RNA conjugates was this paper by Andrew Scott in ACS Chemical Biology, where again, they use this fancy system, and you can ignore everything here. This is essentially just an RNA with a disulfide bond in it, an antibody with a disulfide bond and an RNA conjugated to it. They show good co-localization in cells expressing the receptor to the antibody, and in cells that do not express that receptor, you don't see the green fluorescence in those systems. But interestingly enough, they actually showed that with this antibody-RNA conjugate, with this ARC, with the cleavable bond, you see no knockdown whatsoever. So it doesn't work at all. And it only works, and their postulate was that they were getting into cells clearly, as you can see here, and they had a whole bunch of fact studies showing that they're getting into cells, but they were not releasing into the cytosol. And the way they sort of concluded this was they added chloroquine to it. So again, if you add chloroquine to it, you can start to see knockdown occurring in that system. We've tried this. Chloroquine kills our cells at these concentrations. So I'm not really sure if this is true knockdown, but we'll leave that as is. So again, one of the first ARC systems, the true first ARC systems, doesn't really work. So what do? They actually made antibody-RNA complexes, right? They took the antibody, conjugated it to the 9-arginine, added the RNA to that, and of course, sure enough, that does work, as shown in all those slides before. So again, these antibody-RNA complexes do function, but again, we're still yet to see an antibody-RNA conjugate that actually works. Okay, so the next system here, which I think is the true antibody-RNA conjugate system, was done by folks at Genentech. And this is a thorough paper. I think if you're looking to get into this field, this is the paper to read. This really, they did this in a really nice way. So what did they do? They take RNAs, conjugated to a non-cleavable linker, conjugated also to a cleavable linker. Genentech has this thiomap technology where they can engineer 2 cysteines on the heavy chain of their antibodies site-specifically, so you really only get a DAR of about 1 or 2. It's pretty nice. So they're able to take their modified RNAs that have malamides on there and conjugate it to the antibodies. They show binding. They have really good characterization data, like I showed you before, showing again that these things are conjugated only to the heavy chain of their antibody. Really, really well done. And it's a thorough system. Why? Because they tried a whole bunch of antibodies. And their whole point here was, if we make all these antibody conjugates as a function of pathway, cell line, expression level, what gives? And the answer was a lot of them actually don't work. It's not a good conclusion. But they did find systems that work. And of course, the system that work, for example, this TNB2, which is present on prostate cancer cells, have a 10 to the 6 expression of receptors per cell. So again, things that have super high expressions and high internalizations do work. This goes via the lysosome. It's needed, but it's not sufficient. Why? Here's HER2. Also 10 to the 6, doesn't work. Right? So there are other systems here that have super high expressions that still don't work. So there's something beyond having a high expression in fast recycling that you need to function. These things do work well. Again, at 15 nanomolar, they're not great, but they do work well. They were pretty disappointing in this paper because, again, HER2 doesn't really work. I am way out of time. So I'll skip through this really quickly. What is this? This is another paper where they've done a Fab RNA conjugate. Again, excellent purification, showing that they work. What I really like about this paper is they actually compare themselves using ApoB to the GalNAc system and show that the GalNAc system is just highly superior. Now, this is also because they were using cells that have a lot more acyl glycoprotein receptors than they do have these anti-transferrin, having the transferrin receptors. So maybe that comparison is not fair. But they're also able to make this with a variety of linkers. And what I wanted to get out of this story is they actually found excellent knockdown at 10 mg/kg in calf muscle and heart. And you might ask, why? Well, it turns out in the calf muscle and the heart, you have a super high expression level of transferrin receptors. Even I never knew that. Thought that was interesting. But this system does work. It's a Fab fragment conjugated directly to an RNA. And again, you see activity. Interestingly enough, you see activity via IV, via sub-Q, and also via IP. All right, so skipping through all these slides. Just wanted to finish with, again, this is where we think we need to understand a little bit more about the system. We know that we need these high expression levels, but really it's how they escape and what happens post-escape, or at least what happens post-endosome, that we need to understand. I don't think disulfide bonds are the way to go. In fact, there are folks that understand that disulfide bonds actually don't only cleave in the cytosol, they actually cleave also in the endosome. It's not a well-known phenomenon. It's something that we've been looking at quite a bit in my group, but we do have data that shows these disulfide bonds do come apart in the endosome. And if you're having disulfide released in the endosome and your RNA getting released in the endosome, we actually think escape of just the RNA not conjugated to the antibody is going to be more difficult. And there are data out there suggesting that that's the case. Anyways, I'm way over time. Happy to take any questions that you might have.